Stanford University.
Hi, I'm Bill Newsome. I'm professor of neurobiology at Stanford. I'm also director of the Stanford Neurosciences Institute, and welcome to Open Office Hours. So the Stanford Neurosciences Institute was begun about 1 year ago. I agreed at the request of President Hennessy to direct the institute, and we are dedicated to the proposition that neuroscience, the science of the brain, is going to progress the most rapidly if we integrate perspectives and talent and tools from disciplines as far-ranging as computer science and electrical engineering and psychology, even school of education, in addition to basic neuroscience and clinical sciences like neurology and neurosurgery. And we have got to find ways to bring talent together in new interdisciplinary configurations that are really going to solve and move the needle on fundamental problems in brain science. And we ask groups of faculty to band together across departmental and school boundaries and to actually give us letters of intent. What's your big idea for how we can move the needle on some problem and fundamental problem in neuroscience? We had 22 letters of intent from faculty all around the university. Over 300 faculty were involved from roughly 100 different departments. And of those, we selected 10 to go to the full proposal stage. And of those 10, they were so exciting and so interesting and had so much energy to them. Even though we had only planned to fund 3 to 5 of them for phase 1, we wound up funding 7 of them simply because we just felt like we had to find some way to maintain this energy and this enthusiasm and see what can actually come of this. So enough about me and about the Stanford Neurosciences Institute and what we're doing here. I'm excited about it, but that's not why we're here today. We're really here to answer your questions on office hours. And I really appreciate all of you who wrote in. You've asked really great questions, and whether it came in by Facebook or by Twitter or by email, it's really cool stuff. And the first question that was actually asked and sort of came to the fore, it was actually asked in one way or the other by several people, but people want to know, what are the biggest challenges that you face in neuroscience? And are they scientific? Are they ethical? Are they political? And what are they? What are the challenges in neuroscience? And, you know, honestly, we could sit here and talk for the next hour about this one question alone. We have so many challenges in neuroscience, but, you know, if I had to hone in on a single family of challenges, I think the biggest challenge facing neuroscience right now is how to understand the brain in terms of interacting, functioning circuits. So sets of cells in the brain make chemical or synaptic interactions with other cells in the brain. Certain groups of cells function together in circuits to perform certain computations or to mediate certain behaviors, and those circuits will interact with other circuits to create systems. And how we actually go about understanding the brain at a circuit level and the relationship of circuit activity to behavior and to cognition, I would say that's the biggest single challenge in neuroscience, the single scientific challenge. Now, we say, I've told you now, I think that's the biggest challenge in neuroscience, but let's break it down a little bit more. It's actually worth thinking about carefully. What do you need to know in order to understand a circuit? And I think of this as a table with 4 legs. So there are kind of 4 pillars that the table stands on. And the first pillar is just a structural map. So let's imagine you live here where I do in the Bay Area. And if you want to get from Palo Alto to Berkeley, the first thing you want to do is open a road atlas and just see which roads connect to other roads. I just want a static map. Give me a map, for crying out loud, of how the thing's connected together. But if I want to go from Palo Alto to Berkeley at 4 in the afternoon versus 12 noon or midnight, I need more information than just that static map. You want to get out your Google Maps application or your Waze app, whatever you use, and you want to know what traffic is flowing on which road at any given moment in time, right? We call that dynamics in the nervous system. We want to know what signals are flowing along different nerve pathways. So that's the second leg. First leg is the static map. The second leg is the dynamics. It's the signals flowing within the circuitry. The third leg is that if you're a traffic engineer and you really want to understand how the system of roads and traffic is working, you want to be able to manipulate or perturb the system. You want to know, if I shut down this bridge or I shut down this road, where are the signals going to go after that? How is it going to have system-wide effects, and what's the behavior of the system going to be? So the converse of that is, what happens if I put in a new bridge, or if I widen a road? I pay money to do that. How is that going to change the signal flow within this circuit of roadways in the Bay Area or within a circuit in the brain? How is increased traffic at this point going to influence the behavior of drivers and the overall behavior of the system at other points? So that's kind of the third leg of the table, is this ability to actually introduce perturbations or actually be able to manipulate circuits at a very fine scale. That's the third leg and the third challenge. Fourth one is maybe a little less intuitive, but it's really important, and it's theory. It's what I call theory and computation. So we could make measurements about the dynamics and about the maps until the cows come home. We could collect data for the next 10 years about different perturbations, but unless we develop a theory about what this entire system is for and what its overall purpose is and how the activity of the individual components fit into this purpose and into this overall function, we will never understand the system. So no amount of data collection will help you understand the system unless you have an overall theory about what its purpose is and what it's trying to accomplish. And that's really the fourth leg. That's the fourth pillar. And if you prop all these four legs up at one time and you just let go of them, they're all going to fall over and hit the floor. You've got to have a tabletop on top of those four legs in order to hold this thing together and give it structure and integrity. And that tabletop is behavior. You have to remember that nervous systems evolved to produce behavior. And when I say behavior, I'm being very general about this. It doesn't just mean that muscles have to contract. I include cognitive processes that go on inside the human mind and many animal minds, I think, that actually go on without muscles actually contracting. It can happen long before muscles contracting, but nevertheless influence our behavior. So all of these measurements need to be made. Our maps, our dynamics, our perturbation, and our theories need to be formed in the context of a behaving nervous system that's actually producing its natural product. So I see challenges in all four of those areas. So when we talk about the challenge of circuits, I see it as challenges in developing maps, challenges in measuring the dynamics, challenges in perturbing the system at finer and finer levels, and challenges in developing theories. And the great news is that we're making huge progress on all four of those fronts right now. In my lifetime, there has never been a moment like this one in terms of its potential for an inflection point, a fundamental change in the speed and acceleration of discovery. And that inflection point is occurring because we have amazing new technologies that are allowing us to make measurements of the static map and make measurements of the dynamics and make perturbation experiments that we've just simply never been able to do before. So we're at this point of fundamental change in neuroscience. People like me who've been in this business for 35 years now realize that the tools are so new, the capabilities that we are being given by the tool inventors and tool makers are so powerful that we have to stop and rethink what it's going to mean to be an experimental neuroscientist in the future. And we have to stop and rethink what it means for us to train students. This neuroscientist of the future is going to need to have one leg in neuroscience and another leg in engineering, or another leg in molecular biology and genetics, or a leg in psychology and economics. It's going to be far more interdisciplinary than it has been in the past, and we really have to rethink how progress is going to happen in this field. So on the one hand, we have these massive challenges. On the other hand, we have these fantastic new tools that are going to help us meet those challenges. So I'm extremely optimistic about being able to deal with the challenges despite the extraordinary complexity of this 3 pounds of circuitry that sits between our ears. So the second question that came in, which is really another very interesting question, is do you think that the study of dynamic functional connectivity is going to be important, an important way forward for understanding how the brain works? And the short answer is yes, absolutely. The longer answer is interesting. Remember, and we talked about this in the answer to what are the challenges, when I said what are the challenges. We talked about connectivity, having a static map, and we talked about measuring the dynamics of information flow within the map, how important those two things are. But what this question is raising is a more subtle and important point, which is that the connectivity itself, that static map in the brain, is not really static. The connectivity map itself changes with time, and it changes as people learn, but it also changes immediately during the course of behavior. So if I ask you, for example, if I tell you I'm going to show you a spot of light on a TV screen, if it's red, I want you to raise your right hand, and if it's green, I want you to raise your left hand, you can do that, no problem. But then within 10 seconds, I can say, I'm changing the task on you. Now I'm going to play you an auditory tone. And if you hear a high-pitched tone, you raise your right hand, and a low-pitched tone, you raise your left hand. Well, you can do that. And if you can switch from one of those tasks to the other within seconds, the connectivity, the effective connectivity in your brain has changed. In the one situation, it's visual inputs that are controlling the motor circuits that lead to the hand raising. But in the second situation, it's auditory inputs that are hooked up to and control the motor circuits governing your hand raising. So effectively, the brain is being rewired, and we call that dynamic connectivity. And it can be rewired fast like that. Information flow can be routed in different directions, or it can be rerouted slowly. When we learn calculus, our brains are being rewired in essence. You really are changing the strength of synapses in your brain. So dynamics at fast timescales and dynamics at slow timescales are really important. And it just goes to show we don't have a single map, but the same structure that you look at under the microscope can basically have information flow that flows in different ways in different contexts. And I think the scientist in the United States who has really unveiled that aspect of nervous system function for us with meticulous care over the course of the last 20 years is Eve Marder at Brandeis University, who works in small lobster ganglia that control stomach rhythms. And it can generate one rhythm under one circumstance and another rhythm under another circumstance. And she has shown just beautifully that the system actually rewires. And it will rewire itself. The synaptic connections will change strengths if you drip on a hormone, a neurohormone, a neuromodulator substance called octopamine. And we have lots of these neuromodulator substances running around in our brains, and different substances modulate different parts of the brain. So our brains, many of our circuits in our brain are getting dynamically rewired constantly as we produce behavior over the course of a day. And that's a really critical area for study and for understanding. So it's interesting to ask, and I talk about some of these concepts sometimes like dynamical changes in connectivity, and people look slightly puzzled and they say, well, you know, what does that really mean? How does that play out in practical circumstances? And that's a great question, obviously. And here's the way to think about this. One of the things that really characterizes primate behavior and human behavior is our flexibility. We can adapt behavior to different social situations, and that's a key part of our success in life. So I might have completely different emotional reactions, facial expressions, thought patterns when my boss is in the room. But my boss leaves the room and my wife walks into the room, and the words and sentiments that come out of my mouth, the kinds of issues that I engage, are completely different. These are changes in the nervous system that happen completely unconsciously, but it amounts to rewiring. It amounts to different inputs causing different outputs of the nervous system. And the dynamical changes in connectivity we're talking about is really critical to that socially sensitive production of behavior, which is in turn critical to adaptive life in a primate society. The third question I want to try to tackle here briefly, because it's a whole area of study in and of itself, but the third question I'd like to try to tackle is one that came in that's really important and important clinically. And it's a question about neural regeneration after injury, whether that injury happened from an automobile accident and damage to the spinal cord, whether it happened from stroke and injury to the brain. The question is are there ways that we can encourage nerve cells to regrow after insult or injury in ways that will promote healing, actual behavioral and functional healing from an injury to the nervous system? And this is an extraordinarily important question for lots and lots of people, people who've had injuries or insults of one kind or the other to the nervous system. And I certainly have, one friend in particular who suffers from this, and all of you out there listening, I'm sure, have friends or family who similarly want to know the answers to these kinds of questions. And, the answers are evolving, right? Even right now, research going on right now is changing answers to these questions. Central nervous system damage has always been extremely hard to repair. It's a paradox in the human nervous system and most mammalian nervous systems that if we get damage to peripheral nerves out here in the arm or the finger or the leg, those peripheral nerves will actually regrow. Those neurons have the ability to regrow, find their targets and make connections with those targets, and we can recover a lot of behavioral capabilities for the hands and the extremities after injury. Yet the same injury in the central nervous system, and by the central nervous system I mean the brain and the spinal cord, that same injury, the nerve cells do not regrow. Once they get cut, they just sit there cut. And that's why paralysis in many, many cases is permanent. That's why the effects of gunshot wounds or other traumatic injury to the brain seem to be fairly permanent at times. And the question is, what's the difference between the central nervous system and the peripheral nervous system, such that the central nervous system does not regrow most of the time, yet the peripheral nervous system does? Simple question, right? Well, the first kind of really great experiment on that was done by Albert Aguayo up at McGill University in Canada back in the 1980s, and he did a very simple thing. He had a mouse with a section of the spinal cord. No neurons are regrowing, it's the central nervous system, and he took a piece of peripheral nerve and he just used that to bridge from one side of the cut to the other. And it turned out that the nerve cells started putting out little sprouts and moving across the damaged area along this peripheral nerve, this piece of peripheral nerve, which had the huge implication that there must be little chemicals or little molecules in that piece of nerve that encourage the outgrowth of those neurites and the outgrowth of connections across the gap. And that led to a whole field of neural regeneration over the last 20 or 30 years, which has really been interesting. And some of those molecules that actually encourage growth have been identified, but kind of an unexpected twist of events was that there are molecules sitting there that actually discourage growth. And Martin Schwab and his colleagues at the University of Zurich were really key in identifying some of these molecules. So what you really have in the central nervous system is a cocktail of molecules. You've got a bunch of these growth factors, and you've got a bunch of factors or molecules that inhibit growth, and we're still trying to discover what the various molecular players are. And when we really get that knowledge, the hope is that we'll be able to set the conditions right for regrowth when there's an injury, and we'll actually be able to help people recover function. Some of this is actually into clinical trials in humans right now. So one of the inhibitory factors that Martin Schwab discovered in Zurich is called NOGO. It's a gene and a protein product that's powerful inhibitors of nerve growth. And what Martin showed in experiments in mice and in monkeys is that antibodies to NOGO, so you actually glom onto NOGO and glom it up with an antibody, that will shut down the suppressive function of NOGO, and nerve cells will start sprouting, and animals will actually regain function in their formerly paralyzed limbs. And this antibody treatment to NOGO is actually currently in phase 1 clinical trials sponsored by Novartis. Phase 1 is safety trials, and I think it's passed safety. I think that announcement was made recently. And the next step is to go into phase 2 trials to see if it's actually clinically efficacious, if treatment with these antibodies will actually help restore some function in humans. So this is a very exciting area, and lots of really smart people working on it. And I'm optimistic that within my lifetime, and I'm not a spring chicken, I'm not that young, but within my lifetime, we might actually have real progress on this front. So people frequently ask, well, why are these inhibitory factors there? Don't you want your nerve cells to regrow? I mean, what are they there for in the first place from an evolutionary perspective? Why would you do that? And, I think the answer is complicated, and it's due to the fact that we like some plasticity in our adult brains. If we had no plasticity, no ability to change our connections in the brain, we wouldn't remember anything tomorrow from what happened today. So we have to have some plasticity there, but we don't want nerve cells taking off and growing willy-nilly in the brain, or else our memories that we value would be destroyed. So maintaining some flexibility in learning, but maintaining some stability in terms of memory probably requires this kind of interplay between facilitation of regrowth and synaptic change and an inhibition of regrowth and synaptic change. Okay, the final thing I want to address, we had several questions come in about stroke. And stroke follows right on our theme of regeneration, actually, after injury. So stroke is fundamentally a lack of oxygen to some part of the brain, and nerve cells start dying in that part of the brain. That can be caused either because a blood vessel springs a leak and blood starts flowing out of the blood vessels, or it can be because a blood vessel gets clogged, depriving some region of the brain of its access to oxygen. And this is a really big problem. Stroke historically has been viewed as something for which there's very little hope. People will have some functional recovery, usually in the first days or weeks after stroke, but frequently the effects of stroke are long-term and debilitating. And the question is there anything we can do about this? Can we get any leverage on this? Can we reverse this apparently irreversible condition? And this kind of ties back to some things I said at the beginning of this office hours, that we have this big ideas and neuroscience program going on here at Stanford. And one of the big ideas that about 30 Stanford faculty want to work on is exactly this problem of stroke. They believe that this is not irreversible. They believe that there are some new experiments and new approaches we can take to this problem. And they fall into several different categories. We have some engineers who are really good with devices, and they want to give us a better generation of measurements of behavior following stroke and during recovery from stroke. So they want to put very sensitive pressure gauges on the fingers that will allow us just during everyday behavior to measure the increase in manipulability of fingers and hands. They want to put sensors on palms and see how much pressure people just are able to exert during their normal everyday lives at home, and actually capture all this data by computer to put together a quantitative picture of how people's behavior improves after stroke. And then we really want to do something to intervene. And there are several ideas that are really interesting that are floating around. There's a new important hypothesis that some of the deleterious effects that follow stroke are due to activation of autoimmune responses. So the important B cells in the immune response getting activated in inappropriate ways leads to even more damage and even more neural death is the hypothesis. And if we can intervene at that B cell system in the immune system, we might be able to actually rescue some of the tissue damage that follows stroke. Other interesting ideas are using non-invasive electrical stimulation techniques like transcranial magnetic stimulation, and not simply using it as a diffuse way to exercise cells, but actually using it to figure out which circuits are influencing which other circuits in the brain. So here at Stanford, we've developed a way to combine transcranial magnetic stimulation with magnetic resonance imaging. So we can put in a pulse of electromagnetic stimulation, and using the imaging then, we can see which circuits are responding to that pulse of magnetic stimulation. And the hope is that some of the circuits involving the affected region in stroke, we can actually show are important for creating new plasticity in the area, whereas other circuits may be less important. And if we can actually measure those things with the imaging, we can actually work out the circuits and which ones are best to stimulate in order to restore the affected area of the brain to some plastic state. And that's a cool idea to pursue. And then another idea involves release of some of these excitatory or these facilitatory factors that we talked about, these trophic factors that encourage nerve growth, and blocking some of the molecular factors that discourage nerve growth. We talked about them just a few minutes ago. but actually timing the release of some of these molecules into the affected stroke area through implants that go into the stroke area and then can release these trophic factors over time to encourage local nerve sprouting and local nerve regrowth. And this is a growth area. This is an area where there are really smart people trying to figure out ways to deliver these molecular factors to restore the brain to a plastic state. So I think all of these kinds of things, they're smart, ambitious people trying to figure out ways to do this, and this Big Ideas group here at Stanford may well be among those in the country that are able to deliver real hope for stroke victims. So thank you all for joining me here on Open Office Hours today. If you were really sitting in my office, I'd love to ask you your reactions to this, and frequently people in my office have ideas about some of these things that I haven't thought about yet, and maybe we can engage these some more on a future Open Office Hours. Thank you for joining me.
Stanford University.